Can you see my screen?
Yes.
Yes.
Let me see. This is the online repository on GitHub. And there are several sections. And at this point, I will cover the lecture for molecular dynamics. So this is in the 4 lectures, and then we will go over all these folders during the course. So the first one is MD, and I will use this browser to display the presentation. So can you see the whole screen, the whole presentation, or is it kind of?
It's okay.
Okay, so yes, let me start with this molecular dynamics lecture. The idea of molecular dynamics, it's a bit old. So it started with Isaac Newton in the 1600s. And basically, he was dealing with the interactions between objects. In particular, he was studying the gravitational force and interaction between the Earth and the Sun, for instance, or the Earth and the Moon. So this was a simple two-body problem, right? And this describes the dynamics of these two objects. And imagine that at that time, Newton only was dealing with the two particles, and he was trying to solve this equation, right? And all the computations were doing on the paper, and it was quite difficult. So it was impossible that we could treat several particles at the same time and solve these type of equations. So the basics or the fundamentals of the theory of molecular dynamics is quite old, but now we have novel algorithms to solve the Newton's equations. Besides that, we have a modern hardware like GPUs, right? And where current molecular dynamics code run very fast. And that's why these equations can be solved in an easier way. So this is the Newton's equations. The force is equal to minus the gradient of some function, which we call energy. And this function, U, depends on two things, the particles' positions and the velocities. And because this is a second order equation, we need two conditions. And in this case, I'm thinking that the two conditions belong to the position and velocities. So as I said before, in the case of Newton, he was dealing just with two of these guys here, yeah? Two particles. And now we can treat up to millions of particles, and this is solved by the computer. So in practice, it's not possible to get the exact positions and the velocities of all the particles. So what people do is that they approximate these velocities, for instance, by means of some probability distribution. And that's why, although these equations are deterministic, yeah, and we could get the evolution of a system by solving these equations. In principle, this is not a realistic solution because we will need the exact velocities and positions of the particles. So we do approximations in the simulations. So this is an schematics of the U function, which is the energy of the system. And here in 2 dimensions, I'm depicting the conformational space. So imagine that you are, you start in some position as in this point here, and then you want to explore the entire landscape, right? So in some cases that will be easy because the landscape is flat, right? As in this case, you can just walk and there is no problem. But imagine that you want to explore this hill here, and this will require more effort from your side because this is very steep. And also you need to do that several times. So it's very complicated. This is similar to what the molecules need to do in order to explore this function U, which is the energy. And it's a function of the conformational space. So this is a slide showing the earlier papers in molecular dynamics. The systems that were treated at the beginning were very simple, like the hard sphere system, and then the liquid argon. Then Ariel Washer and Michael Levitt published the first paper on protein folding, which is more realistic. And because of the contributions for these multi-scale models and complex systems, these 3 persons were awarded with the Nobel Prize in 2013, right? And now in this slide, I show you the relationship between experiment and theory and simulations. So some decades back, right? The people were happy with the experiment and also the theory. But now people want to get more insight into the systems, and they are trying to do simulations where experiments and a theory cannot help us to visualize what is happening. For instance, when molecules are interacting, it's very difficult to see them with the experiment. And here, simulations can help. So notice here that when I put this plot, I was thinking about the molecular dynamics simulations, pure computations. But now a big component is being included, which is the field of machine learning and deep learning in molecular dynamics. So because plenty of data has been accumulated over the years with experiments and simulations and theory, and this data is now being processed by means of machine learning and deep learning. So there are different applications of molecular dynamics, and here I will show you some examples. This is a protein, for instance, and it is surrounded by waters, which are these small red things here, and it contains several other components like the ions and some small molecule in the binding site. So this is a picture of adenylate kinase enzyme, and this field is not restricted to biomolecules, but it can be applied also to other systems like clays. So in Umeå, we have several experts in this type of molecules like Magnus Andersson, Magnus Volpast. We have also experts in clay systems like Mikael Holmboe at the chemistry department. So the field is very broad now, and there are other systems like traditionally were not studied with these approaches. And here, for instance, I'm showing you some ice cream research where molecular dynamics is used. And also in civil engineering, people are studying asphalt by means of molecular dynamics. This is very complex because this is not homogeneous and it's a matrix of components that needs to be simulated. So now, how the U function is modeled. So in principle, we don't know this function U, right? And we approximate that by using some empirical potential. And if you look at the literature and you see that some particular force field like charm or Gromos or clay FF is used, then it means that they are using some particular empirical functions or some different parameters are being used. Parameters that need to be modeled in this particular force field are the force constant for the bonds, the equilibrium positions of the atoms, the force constant for the angles and the equilibrium position of the angles, also for the torsions, these parameters here, or for the Coulomb interaction, the charges need to be modeled using some particular scheme. And in the case of the van der Waals, the epsilon, the magnitude of the force of the potential needs to be modeled, and also these sigma parameters. So this is more related to the charm force field. And one interaction is missing for protein, which is the CMAP correction for the Raman-Hundert angles. But the takeaway message from this slide is that the difference in the force fields that you will find in the literature is in this type of approximation that is being used. So this the picture explains what each term refers to. And here is the bond length or the angle type. The bond length is between two atoms, and the angle involves three atoms. We have also the torsion angle, which involves four atoms, and the angle is refers to the middle of them. For instance, this one, this, and this. The Lennard-Jones is one of the long-range interactions, right? And it is for particles that are not close or attached to one bond. And also we can find the Coulomb interaction, which is also a long-range interaction. So this picture is a benchmarking for some force fields like CHARM, OPLS, and they measure here the performance of the different force fields, and they also propose some possible modifications of the force field so that they get a better score, right? So a very common question, what is the best force field? It depends on the type of system that you are studying. And in particular, you will need to look at the literature and see if there are existing benchmarkings for the particular force field. For instance, in this case, we have up to charm 27, charm 22, and they compare with the FF and OPLS, and then you can see if that reproduces the existing data. So it's not something that can be said 100% sure that one force field is the best option with respect to the others. So this is a simple force field or energy function, which involves only the product of two functions, which are the sinus and the cosine. So you see that only with this simple function, we see a lot of valleys and hills. So if you want to travel from one point to another, you need to cross a barrier. And this is what hinders molecular dynamics from exploring the entire energy landscape. Now you see that this function is quite simple and it displays already rugosity. But in the case of the real molecular dynamics, we have a more complex energy function, and we should expect to have more maxima and minima. And that's why it's not easy to get a proper sample in molecular dynamics. So there are different force fields in molecular dynamics. I mentioned some of them: Gromos, OPLS, Amber, Charm. These are very known in the field of proteins and hydrocarbons. There is also a force field called ClayFF for clays. And there is also a Martini force field for coarse graining. Here in my slide, the novel machine learning and deep learning force fields are missing, but they are very, very well, and they are getting more mature. And in particular for these empirical force fields, one needs the parameters of the compounds that we need in our simulation. And if we don't have the parameters, then we will need to use additional approaches like the QM, quantum mechanical approaches, to get the values of the missing parameters. But if the compounds are standard, like 20 amino acids, then we should have the parameters in the standard force fields. So water is very important and illustrates the concept of the parameters. And there are different models for water. We will be using this three sides water model, which is called the T3P in charm force field. And here you see that in the three-side model, we need three particles and the parameters that describe the interaction. There are also more elaborated models and more accurate, especially in the field of spectroscopy, where you include more sites on the atoms. But because water dominates the simulation box, they are the most expensive part in the simulation. Now imagine that you include 2 additional atoms in the water. So the simulation becomes heavier, and you need to consider that, but this could be also more accurate. So we will see in this course a simulation with amino acids. I will not discuss other systems like a clays or a matrix of components. So we will study standard amino acids. We have 20 amino acids in nature, and they differ in the type of side chains that they could have. And here there are the possible side chains of the 20 amino acids. So for protein systems, there are already available tools. And in particular, the Protein Data Bank provides a lot of information, including the crystal structure, for instance, in this case is adenylate kinase. And then you can model the structure of the protein by using this set of coordinates that you get from the Protein Data Bank. So in our simulation, in general, we can include only a molecule like a protein or some small molecules or compounds plus some solvent like water, right? For the simulation, cannot be very realistic in the number of particles because currently we can achieve, for instance, up to millions of particles, right? But in a wet condition, right, we can have up to 10 to the 23 particles in the experiments that the people in wet lab does. And in order to simulate this type of conditions, right, with many more particles than in a realistic simulation, we use the so-called periodic boundary conditions, where the initial cell, which is this one, is reproduced in all directions, and then the particles are allowed to interact within the so-called minimum image convention, right? And using this periodic boundary condition, we can mimic somehow a more realistic system with this number of particles. This is a trick that is used in molecular dynamics. Here I display again the system for the enzyme. You see that this is the box that I'm using, right? And it contains only 30,000 atoms. But if I use the periodic boundary condition, then I will mimic a more realistic system. So a very important component in molecular dynamics is the electrostatic energy. And this is tricky to compute. And in particular, one algorithm that is used is the so-called Ewald method, where this summation for the computation of the Coulomb interaction is split into a short range interaction and another long range part. So we will use the Ewald method in the particle mesh Ewald flavor in our scripts. So there are different ways to integrate the Newton equations. And here I show you just the simple leapfrog scheme where you compute the velocities at the midpoint of the interval, and then you update the positions, and also you use the midpoint velocities to update velocity, the velocities at the next interval of time. So this is just one way to integrate the equations. There are several, and just an illustration on how you can solve the Newton's equations. Ergodicity, this is the property of the system that will allow us to compute average of certain properties. For instance, imagine that I need to get the average motion of the atoms in a system like this one, which is very big. And then I can follow the trajectory of the particles along the time, right? So I can take a snapshot of along the time, or I can only take snapshots of this system, like if they were pictures or some configurations of the system. So in this way, we can have the average properties of the system we are studying, and this will allow us to compute that with molecular dynamics, because in molecular dynamics, we have the evolution of some operator or some property, and then we can get the average by taking snapshots along the time. So again, this is the conformational space that we will be exploring. And in the ergodicity, we are sure that we can sample all this landscape, right? If we leave the system to progress for a long time, right? And this is the basic principle of molecular dynamics. But the problem can be that in practice, as I said before, it could be very difficult to get over this hill here, right? With the standard molecular dynamics. And although ergodicity is valid, then in practice, we cannot get over this hill. And that's why we will see tomorrow how to use an enhanced sampling technique that will allow us to cross over these big barriers. So if you read the papers, you will see that there are some abbreviations like MVE, MVT, MPT, and so on. And it refers to the different ensembles that you can see or you can obtain in molecular dynamics. MVE refers to the microcanonical ensemble, where the partition function is given by this expression. And this stands for a constant number of particles, volume, and energy. And this partition function is very important, and maybe the most important thing, because based on this expression here, you can get all the thermodynamical properties of the system. For instance, here is the entropy, right? Which is in a MVE ensemble, is just a logarithm of the partition function. We can have also the canonical ensemble, in which the constant number of particles, volume, and temperature is maintained. And the partition function is given by this expression here. And just as an example, you can see how the free energy is computed. Yes. There are other more elaborated ensembles like the MPT, which is where the number of particles, the pressure, and the temperature are kept constant. And also the grand canonical ensemble where the number of particles is expected to change. So in order to keep the properties constant, we need some particular thermostats or barostats. And for instance, in the MVE, we are keeping the energy constant. In MVT, we are trying to keep the temperature constant, and this can be achieved by using different thermostats like Berentzen, velocity rescaling, Nose-Hoover. This Berentzen is useful just for initial equilibration, but it shouldn't be used for a real production. And there are also other properties that need to be kept constant. For instance, in MPT, the pressure needs to be constant. And here, the barostats can be used. And in particular, these are common, the Berentzen and parallelogram. So we saw before that in molecular dynamics, we need to solve Newton's equations for millions of particles maybe. And this involves a lot of numerical computations. So that's why I mentioned that in the times of Newton, Galileo, and all these giants, they could only solve for a two-body problem, for instance, on paper and pen. So now we can solve that for millions of computations in a second. So how can we speed up these computations? This is one schematics. And here we see that there are some people on a lane and they are trying to pay. And there is a cashier that will receive the money as sometimes when I buy in the grocery store, a big lane is formed. And then when another cashier comes, then the lane is split into several lanes, and then the time for paying is reduced completely. So this is called parallelism, and this is how the Newton's equations can be solved in a faster way, where several computers solve one part of the problem, and they are done at the same time. So by using several computers, we can even increase the size of the problem. So if you are trying initially to simulate a problem with 1,000 particles, and we increase the number of computers, then we can solve maybe 100,000 particles, right? So we can get the calculations faster and also increase the size of our problem. In our center, we have the cluster that is called Kebnekaise, and it looks like this when the lights are off. And this is the machine that we will be using today. There are different techniques to increase or to speed up the simulations. For instance, a different type of parallelization scheme like MPI, OpenMP. In these parallelizations are the ones that are included, for instance, in NAMD. Algorithm techniques are also included. For instance, different types of molecular domain decompositions, right? And this is just an illustration or a schematic of how the molecular dynamics code looks like. And here you see that there are some number of particles, and the position of the particles is updated by means of the force, right? This is just one way to update the position. And then if we perform this computation on a single core, it will take maybe a long time, depending on the number of particles. But we can also split this into several processors, and then the simulation will be faster. This is just a schematic of how the architecture of Schematics looks like, or how the architecture of a machine looks like in a modern computer. Regarding the algorithms, we can also find in the literature currently coarse grain simulations, which are more mature in those days. And here, for instance, instead of dealing with all the atoms in the system, we take some interactions considering a group of atoms, right? So in this way, we'll reduce drastically the number of atoms, and the simulation can run for a longer time scale. So this helps us to study complex systems like this system here, in which each of these filaments is composed of different proteins, and the proteins can be reduced in the number of atoms. Otherwise, imagine that you need to deal, each bit of this is composed of this type of system with maybe hundreds of atoms each. You won't be able to simulate a system like this. So in this picture, I illustrate how the fields of simulations, molecular simulation looks at this point. And here, for instance, there are different types of techniques that I use in computational chemistry, like ab initio simulations, semi-empirical, and classical dynamics. We will be using this type of simulations in the present course, right? And this can achieves more than 1,000 of atoms, for instance, and it can also help us to see the different conformational changes in the structures that we are using. And the time to scale here is beyond the nanoseconds. But a more accurate methods like ab initio will allow us to deal up to maybe hundreds of atoms, and the simulation time scale could be picoseconds, and simulating nanoseconds would be a endeavor. Software, there are different software that are available, like NAMD, GROMACS, Amber, Charm. So today we will focus, today and tomorrow, we will focus on NAMD. And this will be the steps that we will follow in our exercises. And this should be done in all the molecular dynamics schemes. First, we need to set up the system, then minimize the system to avoid atomic clashes. Then we solve the system, neutralization, equilibration, production, and then the analysis. So minimization can be done at different steps to reduce the clashes of the atoms, for instance. Okay, is there any questions at this point? ID, right? So what I will do is that I will show you how to get the exercises that we'll need. So first of all, in the website of the course for the GitHub repository, we give you some initial instructions, right? It says move to the project directory. And this is what I am doing here. Is the size of the text in the terminal good enough? I mean this one.
It's a little bit small for me, at least.
Let's see, let me then increase it. Zoom in.
Okay, I think that's fine.
Okay. And so if you haven't done it, well, I skipped one step. And this was opening the client for ThinLink. And I will close this. If you open the client, you will see this box here. And Brigitte mentioned that some options need to be unchecked. Like in display, you should check this window, right? And then here you type your password. I will type mine. Wait a minute here. Then after typing your password and your username for HPC2N, you will get into this environment. Then I'm using the main terminal that can be obtained in system tools, and then in this part, which is called mate. So what I did at just the first time was to drag this icon to the desktop so that I get it every time that I log in. So then open this terminal and then type CD and the path to the folder that I mentioned already. And then you will be located into the folder. So here you type the username with make dir, and this will avoid that copying the repository will be available to other users, right? After that, move to your username and then type this command git clone, and then move to the folder name for the course repository. So I did that already. And here you can see all the files that I have in the repository. And these are the lectures and the different phases of the course. So I will start with the 0.9 NAMD. And these are the different files that are available in the directory. Also, if you want to get a graphical view or a more graphical view, then you can go to the online repository. And here, the README file will be displayed in some render format so that you can see what is code and what is text, like in this case here. So for NAMD, we will need some files. We will need a PFS file, which is for the structure and some parameters of the structure that we are studying. Besides this structure file, we will need a file for the coordinates of the atoms. And in this case, I'm using this file. And the test case for this example is just a water box with a solvent, which is water. And the parameters file is from the transfer field. And these are the parameters that I mentioned for the force constants, for the torsional parameters, and for the non-bounded parameters like van der Waals. So these are standard files that you can get from the developers of the force field, like the Mackerel. In one of the exercises, in the first one, I put a link for the Mackerel's website where you can download several force field parameters. So for running NAMD, I begin to show you how to run a CPU version for MPI NAMD, and also there is a possibility to run it on GPUs by using the non-MPI version. So notice that the non-MPI version runs only on a single node. And then we can analyze the trajectory in different ways. I will show you some of the ways that we can use. And some useful thing is how to look at the number of nanoseconds per day that you can get in a simulation. And I included two scripts. One which was downloaded from the NAMD website, which uses Python, and I translated that to Julia in case you prefer to use Julia. And these scripts are included in the repository. So how can I run these scripts? So first, let me do a ML purge. And Birgitte mentioned that this is a good practice. So here I'm using ML purge. I'm not redirecting the output to the def null. But in the scripts, you can do that. It makes the output clearer, neater. So first of all, let me visualize the system that I have here. And you can use that, in this case, VMD. So let's type ML Spider VMD. And you will see the different versions that are available. I will use this in the present case, but you can use the other one as well. And I will look for the prerequisites of VMD. And these are the two prerequisites that I need to load first. Before loading VMD. Now, let's look at the system. And I will open VMD and include the structure file and the file of coordinates of the system. So in this case, you will see a box, right? So I can use the orthographic display. And here is a box of water, a cubic box. And then the system of interest in the present case is just the ions, which are NCl and ACl ions, right? This is a water box, so just with 2 ions. And then if I use, for instance, if I type the number 2, then I can measure distances, right? And this will be very helpful. So this is my system, and the best practice is to first explore the system and how it is composed. And we have seen how to load a VMD and the dependencies. So now let me open a new tab. So if I go to File, I will open a new tab. And I can increase the size of the font, okay? So let me go through the files that I have in this repository. Here is the Instructor file, and this is the file that we need to generate in the exercises. And there are some standard scripts and workflows that you can use for proteins, for instance. But if your system is very new, especially in the clays or in the material science field, then most probably you will need to do that manually, right? But in the case of biomolecules, there are already standard tools to generate that, and the exercises will show you how to deal with this. So this file shows you how many atoms are in the system, and also the type of atoms. Here is an oxygen and hydrogen. And for instance, you can see the bonds that are formed between the atoms, right? So if there is not an available tool to generate this file, you will need to do that manually, right? Following the standard format of NAMD. But most probably you can do this on the on the VMD terminal. So this is the structure file. Then we have the standard PDB file, which is just for the position of atoms. Here you have the type of atoms, and these files are the coordinates. These columns, sorry, are the coordinates. Then you have the occupancy and the beta column. And this is the segment name. So for NAMD, you need a configuration file, which will tell NAMD what to do. And here I'm setting some parameters in case you want to restart the simulation or to run that for the first time. And here I instruct NAMD to open the protein structure file that I showed you. And here the coordinates file, which will be the PDB file. Some parameters for shape, the time step. If you want to fix atoms, the number of time steps that this file will run, and this is important for the course, the output name of the file. This will be the string with which the output files will start. The temperature of the system, and this is a flag for setting the Langevin thermostat that I mentioned in the lecture. So this is just an scheme for how to restarting the simulations, but you can also use your own scheme. Here you set the time step, and notice here that this is a variable, right? This is the variable that we set before. This one. And variables here start with the dollar symbol. Then we can use the parameter file. In this case, I'm choosing the charm, but you can use other type of parameter files. And these are some parameters for the non-bounded interactions, including the cutoff, the pale list distance, and the switching functions, which are used here. Also, for integrating, you need a time step, which was set up above, and the shake algorithm for constraining the atoms in the relative positions. And these flags here are for the non-bounded frequency updating, right? And this is used for electrostatic interactions. This is the part where we put the value of the cell vector. And here there are, because the cell is cubic, we have 3 directions that need to be defined. And because all the lengths are the same, then we can fix that to the same value. And I put this in 40.3, which is the size of the cell. But here you can get more complicated shapes like RHDO, for instance, or other type of cells. And what will be different is the values of the cells vectors that you will get for these shapes. But for the present case, it's a standard cubic shape. So this is just a scheme for wrapping the waters, and this is for the particle mesh equal for the column interactions. This part says that the thermostat, which in this case is Langevin, and here is the barostat, which is also a Langevin piston with some parameters. The output files will start with the string that I mentioned before, and the binaries are written. And these are the frequency where you will write the restart file, the trajectory file, and the output for the energies. And here, at this point, I will tell NAMD run the simulation for n steps. And n steps what was set previously, and it is just 15,000 steps. So we'll run the scripts on NAMD using the SLOOM batch system. And in the particular case, for the CPU, we will use the following script. So we have a project, as Birgitte mentioned before, and the project has this number. So I need to fix it. So the script will run for 10 minutes. It will use one node. And then I am requesting 28 tasks. So in addition to this, if I want to use the reservation, I need to add this line here for the reservation and for the day one. So notice that Brigitte mentioned that because we are using pure GPUs, the nodes, then we need some additional flags in this script. So although I'm not using the GPUs for this run, yes, I need to add these lines because the nodes have a GPUs. So this is one consideration that you need to take into account, right? So let me do this. And then I submit the script with the sbatch command. And then I get a number which is called the job ID. So then I can use the command sq -u and my username, which in this case is Ojeda-May. So I see that the script is running, it is here, and the job ID is here, right? So the good practice is to use the job usage command and the job ID. You will get a URL, so I can get this URL and copy, then go to my local browser, and I will paste this. So then here you can see how the resources are used for the particular script, right? So you can see how NAMD is using the CPUs, and also in case that you are using the GPUs, then you will see some number here. But at this point, because the script runs only on CPUs, you won't see anything here. So this can give you an idea on how the resources are being used. So also, I can use the following line, I minus FN 400. You can change this number here and the output. And here you can see how the simulation is running and get some values, right? And something that I mentioned as well in the repository is that you can use the scripts for getting the number of time steps. So I can do this step here. I will purge. Then I load Julia. And then I just run the following step. Julia, the name of the script, ns per day.gl is the Julia script, and then the output file name. And this will give you the number of nanoseconds per day. So if you don't want to use Julia, then you can use the Python script. And I will purge again. And Python is the Python.7 version that is available without loading any module. And because of that, you can use Python ns per day.py. and the output file. And this will give you the exact same answer. You can use this script to get the number of nanoseconds per day. And if you remember, we fixed the string for the output file names. And because of that, all the output files start with this particular string here. Right? So this will be very important because if at some point you restart the simulation and use the same output file name, then you will overwrite the trajectories and everything else. So here I have the outputs, and what can I do with this? So I will move to the tab where I have loaded VMD previously. And then I can open VMD now with the name of the PSF file. And then I can also load the initial coordinates file, but this is not necessary. And now the important file, this step, is the DCD file. Yeah. If you open this, I can use the orthographic display. And then open this DCD file, you will get some number of frames. And if you run this, you will see how the system is evolving, right? So at this point, I'm not interested really in the waters, but I'm interested in the ions. So I can select the representation for ions only. And then I will use a CPK. And if I press a Control R on VMD, then this will center, yes, the system that I selected. And pressing the number 2 will put me in a distance mode calculation. Yeah. So some useful things or plugins from VMD can be obtained here in this menu bar. So if you go to display, no, if you go to graphics, sorry, and then labels, then you can choose here bonds, and you will see here the distances that I have selected. In this case, it's just a distance between CL and NA. And then I can go to graph. And if you graph this, you can see how the distance between these two atoms have evolved, right? This is just one plugin from VMD and some analysis tool. Another thing that you can do in VMD is to use the extensions. And this will be used in the next step in the exercise. And here you can see the different analysis tools. Here, for instance, you have a tool for computing the root mean square deviation for a trajectory. And we will also see today and tomorrow the quick MD tool. So, okay, so it seems that this is is not working. But let me try the next. So, okay, it seems that it's not working with the version 3.8, but let me try a different version of VMD. So I will purge. Then I will search for VMD. I will load. I will open first VMD and go to extensions, no, modeling and quick MD. Now here it works with the version 2.7, yes, 1.9.4 and Python 2.7. So this is the window that you will use in the quick MD exercise. So, okay, do you have any questions about this before moving to the exercises? In the exercises, you will follow the steps. And here, for instance, you can start first in the first part with the TK terminal setting for VMD. And you can follow these steps here and get the trajectory and analyze the trajectory by using the standard tools, like here in extension of the one that I show you for computing the distances between the atoms. So is there any questions at this point? In the first exercise, the purpose was to see how the TK terminal works, look at the steps that you need to follow on the terminal to set up the system, right? And this was the purpose of this first exercise. At the end, you should get the trajectory. Yes, and then you can do some basic analysis, like computing the RMSD along the trajectory. In case you don't know how to compute this RMSD, before I go to the second exercise, I will show you quickly. So you have, I will go to my first exercise, which was the simple case for NaCl. Then here, you can go to analysis, extensions, analysis, and RMSD for the trajectory. And here it will ask you what atoms do you want. The default value is protein, but here I can choose, for instance, other atoms like ions, or if I have a protein, just a protein. Here you can use the backbone and so on. But let me see if I can get this for ions. This will be new for me. I haven't tried this. And here, before computing the RMSD, you should align the structures and computing the RMSD. So if you want to plot it, you can plot this and you can see how the RMSD evolves. Yeah? So, okay. So this was the first exercise. And now let me go to the second case, which is a similar case for a protein, but in this case, you will use the quick MD plugin from VMD. And then you need to follow the steps, and you will see that in this case, you don't need to type commands as in the terminal. This is more convenient. For instance, you have several systems that are similar, and you need to change, for instance, just one residue, and you mutate the residue, or you change some atoms, and then you want to run the simulation in a more standard way or easier way, so to speak. But as I said before, with this tool, you cannot set some systems, like for instance in material science and so on. It's because this is more for biomolecules, and it's prepared for biomolecules. That's why the TK terminal is more flexible, I would say. So the goal is the same, to get the trajectory and to analyze that with VMD. So these are only two ways to set up the system for NAMD. And those days, there is also a tool which is online that is very convenient, and it's called Charm GUI. So before, you didn't need an account for Charm GUI, but currently one needs an account, right? And then you can log in here. And in this case, you get this you get this interface, and these are the possible options that you can use for setting the system. And in the present case, for instance, you can use a solution builder for getting a solvated system. So the protein that we studied in the exercise 1 was the 4-AKE. So here just type the protein ID for the PDB data bank, and then you proceed in a graphical way, right? So this is very convenient, and at the end, you will be asked if you want to set the output in the GROMACS or NAMD or Amber output file layout. And this is very convenient, but there are several cases where this interface doesn't work for very well-behaved proteins and crystal structures. That works, but it is not general. But if it works for your system, then it's a good way to work, because here you can do several things like mutations, changing the ion concentration and the box size and so on. So it's very convenient. Otherwise, you can work with the settings that I propose for QuickMD here in the second exercise, or if the system doesn't behave very well, then you can go to the TK terminal and use the tools that I mentioned. Okay, you can start with this exercise, and please, you can ask any question at any point or write it on the document.